Hi, my name is Bethany Sims, and this is my gut microbiome project. I'm in section 006 of BioLab. Okay, so the abstract. So basically, the gut microbiome influences the health of an organism. It is highly correlated to health, diet, weight, and many other things. So our experiment basically was trying to figure out whether a small dog or a big dog would have a more biodiverse had more biodiversity in their gut microbiome. And so our hypothesis was that bigger dogs would have more diversity due to them consuming more food. And we had 4 samples. These samples consisted of 2 German Shepherds and 2 Yorkie Poodles. So basically like 2 big dogs, 2 small dogs. However, our hypothesis was not supported in this experiment as size does not matter in gut microbiome diversity. Okay, and so our introduction. So just to backtrack, to define a gut microbiome, it is basically the type and proportions of microorganisms living in a given location. The gut microbiome is defined as a large collection of microorganisms found in the gastroesophageal tract, which plays a role in our health. And so the question of our experiment is once again, will a small dog or a big dog have more biodiversity within their gut microbiome? And the hypothesis of our experiment is that bigger dogs, such as German Shepherds, would have a more biodiverse microbiome since bigger dogs, such as German Shepherds, consume more food. Okay, so up next is our introduction. Our methods and materials. So the first step is to purify the DNA from the fecal matter, and this is done with the Quagen kit. And the main goal of this step is to isolate the DNA from the fecal matter. And then following the isolation of DNA, we then concentrate the DNA with the Zymo kit. And this step is significant as it results in a much more concentrated DNA, as the DNA is concentrated to tenfold. Then afterwards, we quantify the DNA using a nanodrop spectrometer. And basically, a nanodrop spectrometer measures the absorbance at 260 nanometers and compares it with 280 nanometers in order to tell the abundance of protein and DNA in a sample. And I'm going to go to our results. So our nanodrop results were pretty good. Pure DNA ideally should have the absorbance of 1.8. And so for our samples, sample 643 had an absorbance of 1.84. Sample 406 had a sample of 2.35, which is pretty high. Sample 1048 had an absorbance of 1.81. And then sample 512 had an absorbance of 1.76. And so most of them had pretty good absorbance other than sample 406. And then, so after the quantification of after the quantification of DNA, we then proceeded to the next step, which was the amplicon of PCR, which amplified regions of the 16S ribosomal RNA gene. And this gene was very important as it held a lot of valuable information. Yes. And then so PCR, basically it occurs in 3 different stages. The first stage being denaturing, which is when the temperature is raised to 95 degrees Celsius. Afterwards, the next step is annealing, which is when the temperature decreases to 55 degrees Celsius. And then following that is extension, when the temperature increases to 72 degrees Celsius. And the number of cycles conducted during this cycle is 25. And then following PCR is gel electrophoresis, which is done at 250 volts. And so the purpose of gel electrophoresis is to check the products of the PCR reaction. And so here are gel electrophoresis results. The two primary things that you should look at here is the positive control and the negative control. As you can see, the positive control is very bright and vivid, which is good because it indicates that there's a lot of DNA in this sample, which was pretty ideal. And the negative control, as you can see, was very, even though it was faint, you can still vaguely see it, which is a bad sign because the negative control was supposed to be completely transparent. And so we believe that cross-contamination had a factor to play in the fact of why the negative control showed up. And so following gel electrophoresis, we conducted PCR cleanup with the Ampere kit in order to clean up the amplicon from the PCR reaction. And so during PCR cleanup, primers DNTPS and PCR fragments are cleaned up. And then following PCR cleanup is indexing PCR, which adds a unique set of tags to identify which sequences came from which sample. Which sequences came from which sample. So indexing PCR pretty much gives each sample a unique set of tags to help identify which sequences came from which fecal sample. And similar to amplicon PCR, indexing PCR also occurs in 3 different stages. It occurs in denaturing, annealing, and extension as well. And each stage has the same temperatures as the previous step. As amplicon PCR. So 95 degrees Celsius to 55 degrees Celsius to then 72 degrees Celsius. And indexing PCR involves 8 cycles rather than 25. And so following indexing PCR is again PCR cleanup with the Ampere kits. And so the purpose of this step is to clean up the Mg2+, the DNTPS, and the enzymes and the reverse primers. And then finally, the last step is to quantify the DNA with the fluorometric assay to measure concentration. And the fluorometric assay was used rather than the UV spectrometer because the fluorometric assay was much more precise. And so here are results from the fluorometric assay. Our results are very low compared as the concentration should be 1.6 UG. Rather, our concentration was very, very low compared to what we were supposed to get. And this was likely due to us not having enough DNA to correctly measure concentration in these samples. Okay, and so here are our sample readings. So sample readings should be about 5,000 total successful readings, and as you can see, all of our samples meet this requirement except for the negative control. Our negative control has a total of 18 successful readings. However, that's considered normal since it is the negative control. And so moving on, this is the kingdom results. As you can see, basically all of them are 100%. And then here are our order results. And then moving on to the genus results. Here's the genus results of the positive control. The genus results of the genus results of sample 643. The genus results of sample 406. The genus results of sample 333. And then the genus results of sample 342. And so discussion. So basically, in conclusion, let me go back to the nanodrop. So to start off with the nanodrop, the extraction of the DNA was proven successful by the UV spectrophotometry. Sample 406, as I mentioned before, had a pretty high absorbance. However, it was still considered fairly normal, as the normal range for absorbance is between 1.5 and 2.2. And we kind of inferred that sample 406 was likely contaminated with RNA. And then moving on to the gel, to the gel electrophoresis. As mentioned before, the positive control was the brightest, and the negative control still showed up despite it supposed to be clear. And as mentioned before, this was likely due to cross contamination. And then moving on to the fluorometric assay, the fluorometer results. So, as mentioned before, there was likely not enough DNA to sequence adequately the concentration. And also, there's likely cross contamination when getting these results. And then finally, we already flipped through these the genus results. However, I wanted to stop at sample 333 just to point out the abundance of bacteria in this sample. It basically just indicates that this dog, which was a Yorkie poodle, had the most bacteria, as it had 8, compared to all the other dogs, which had less than 8. So it kind of proved that the Yorkie poodle, well, it proved that small dogs could have a very diverse gut microbiome as well as big dogs, and that size does not play a factor in diverse gut microbiomes. And basically, that's the gist of my project. Thank you for listening, and have a good day.